Have you ever had the misfortune of getting a poisoning from a meal or a drink that seemed perfectly fine? From Salmonella in eggs to Listeria in fresh produce, undetected pathogens don't just threaten public health, they lead to massive economic losses and food waste. A significant portion of the world's food supply is lost due to spoilage, where the culprit can oftentimes be the same bacteria responsible for foodborne diseases, but also microorganisms such as lactic acid bacteria, yeasts, and acetic acid bacteria. Needless to say, effective surveillance of pathogens in food and beverage, along with the detection of spoilage bacteria, is critically important. Traditional culture methods can take multiple days to confirm contamination, meanwhile a batch is waiting in a fridge, moving closer and closer to the spoilage date. Rapid detection helps regulators and producers act before contaminated products reach consumers. Molecular techniques such as PCR have revolutionized this field by offering detection in hours or even less.
What is the biggest challenge? The food itself. Food matrices are notoriously difficult to analyze. Fats in milk, organic acids in produce, and chemicals in drinking water act as inhibitors that can fail the detection of target amplicon. Also, detecting a handful of cells among millions of harmless microbes is extremely difficult, which is why faster methods can sometimes miss low-level contamination. To overcome these pain points, scientists use highly sensitive molecular assays that can target DNA or RNA of the pathogen directly. A collaborative study by European research laboratories demonstrates that multiplex real-time PCR provides rapid detection of specific bacterial targets, compared to traditional culture-based methods, which may require several days. High-resolution melting analysis performed on the light circular allowed discrimination of 6 different genotypes based on distinct melting peaks. Food matrices are also often analyzed for adulteration. In a study conducted by researchers from an institute in Brazil, cow, goat, buffalo, and sheep DNA were successfully quantified in milk and dairy products using qPCR. The method achieved a very low LOD of DNA for all 4 species, enabling detection and quantification of adulteration among these dairy sources. The success of multiplex pathogen detection from inhibitor-rich samples depends heavily on enzyme performance and reaction chemistry robustness. Solis Biotine's products performed immaculately in the 2 showed examples, as well as in many other academic studies or routine testing settings.
Our hot fireball DNA polymerase is the gold standard for these complex food matrices. Because it is a hot start enzyme, it stays inactive at room temperature and it only activates at higher temperatures, ensuring that your primers don't bind to the wrong targets, which is crucial when you're multiplexing pathogens with high concentration spoilage bacteria. And with Solis Biotine's stability tag technology, these mixes are stable at room temperature for 30 days. This means consistent results even in field labs or regions where cold chain shipping is unreliable.
Every year, millions of people fall ill from foodborne diseases. We just discussed how Solis Biodine mixes can handle the high DNA background from food samples while maintaining the high sensitivity for even a few cells of E. coli or Salmonella. With our reliable PCR products, we are proud to contribute to a safer, well-controlled food industry. From Estonia, we ship worldwide. Reagents manufactured in Europe put to work all over the world. Use this promotional code to order your free product sample.